Myself, Dr. Jibran Ahmed, presents to you Simply Pathology, and today we are back with a very important lecture. So today we are going to read about the theory of staining. Now let me just share a small story with you before I proceed with today's lecture. So once there was a resident who was sitting for the MD pathology exam, and basically in the exam, he had one section that was the tissue processing or the basic histopathological technique section. So over there the examiner had asked the student to perform the H&E staining. So the student had very nicely, he had performed the H&E staining, and then he had gone to the examiner to show the particular slide, and he had entered the viva. So in the viva, the examiner had asked him certain questions. What were the questions? The examiner had asked him that what is the basic theories of staining, or what are the mechanisms, name the mechanisms of staining. Then the student did not reply anything. He did not know the answer. Then the examiner asked him, what is a mordant? Again, the student, he could not answer. Again, the examiner asked him, what is the meaning of progressive and regressive staining? Again, the student could not answer these questions. Instead, he started telling the steps of hematoxylin eosin staining. So the examiner was not very happy with the student. So ultimately the student had passed, but he could not score very well. So what is the moral of the story? The moral of the story is that in your exams, your basics has to be very strong and very clear. So these questions, what are the mechanism of staining? What is a mordant? What is progressive and regressive staining? These are not very hi-fi questions. These are very basic and simple questions that you should be able to answer in the exam. So with this background, we are going to start today's topic of discussion, that is theory of staining. So basically, tissue processing. So we had read about the tissue processing in the previous lecture, wherein we had read about the different steps of fixation, dehydration, then clearing, then paraffin embedding, followed by the microtomy. Now once you have taken the tissue section, that is colorless, that is colorless. You cannot see anything through the tissue section. Why? Because the refractive index of the fixed protein and the glass slide, it is the same. So that is why the tissue section that you have taken, it is colorless. So you need to stain the particular section, so as to view the morphology, you have to stain, and that is where the role of the different dyes come in. So dyes, basically they have specific affinity for different tissue proteins, and therefore they color them differently. So this helps us to understand the morphology of the tissue. So very important, to make out the morphology of the tissue, we have to use dyes. Now if you look at the classification of the dyes, basically there are two types of dyes. One is a natural dye, one is a synthetic dye. So natural dyes, they are derived from plants and animals, and they are rarely used. So one exception to this is hematoxylin and carmine, because they are yet widely used in today's world. Most of the dyes, they are synthetic, and they are derived from petroleum, so they are a petroleum derivative, and most of the synthetic dye, they have a central benzene ring which is colorless. So if a chemical group is inserted in this particular benzene ring, it changes into a colored compound called as quinone, which is actually a colored product which is acting as a chromogen, that is giving color. So to explain this concept in detail, let me just show you this particular chemical structure so that you can understand. So normally the benzene, it is colorless with the formula C6H6. Now if there is basically addition, if there is a modification and addition of certain groups, for example over here this hydrogen is replaced by this oxygen atom over here, as we can appreciate, and once this is occurring, this benzene, which is a colorless compound, is converting into a compound called as quinone, which is a chromogen written as C6H4O2. And this compound is basically colored because of the chemical modification. The hydrogen is replaced by the oxygen, and these groups which are participating in production of this colored compound, these groups are called as chromatophore. So I hope this is very clear to everyone. So the grouping which is imparting the color, they are called as active chromatophore group. So there are two types of compound. One is the dyes can be natural or they can be synthetic. Okay, so how does a particular dye produce color? So how is it that a dye is producing color? Let us try and understand this concept. Now remember, every dye, okay, they are having two components. The first component, it is a chromatophore, and the next component or the other component is the oxochrome. Okay, so basically remember that normally the white light, okay, the white light in the wavelength or in the range of 400 to 650 nanometer is containing all the 7 colors. That is, I am talking about the Vibgyor. Okay, so white light in the wavelength of 400 to 650 nanometer range is containing all these 7 colors. Okay, so as I told you, the dye is having two components. One is the chromatophore, one is the oxochrome. So just let me make you understand that how does they produce color. Now for example, this chromatophore, okay, that is the component of the dye, it is basically observing light of a particular wavelength. Okay, so over here it has observed both, it is observing a light of a particular wavelength. Now whenever the light is incident on it, for example, it is absorbing the red light as well as the blue light. Okay, so and it is completely emitting the green light. Okay, so understand, so it is basically absorbing a light of a particular wavelength belonging to a particular group of color. Out of those group of color, it is absorbing certain groups, okay, and it is emitting the rest. So the color over here will come out to be green in color. Understand? So this is basically what a chromatophore does. Okay, it is absorbing the color of a particular wavelength and then emitting the rest of the light. Okay, and whatever light is getting emitted and falling on the naked eye, the color of the dye is becoming that only. So this is what is the basic concept of how a dye is producing a color. Then I also told you that a dye is also having another component that is called as an oxochrome. What does this oxochrome do? It is just a helping hand. It is actually helping the chromatophore to absorb more amount of light. Okay, it is helping in greater absorption, and it helps to intensify the light, intensify the color. Okay, so how does how does it participate in helping the chromatophore? By donating the electrons, as we can appreciate. So let us read whatever we have discussed right now. So the chromatophore, if you see, they are absorbing light of a particular wavelength, okay, of a white light, okay, representing a specific color, and emits light containing rest of the color, which is giving the color of the dye. So the color of the dye is whatever color that has been emitted. Okay, for example, in this particular example, as we have read, example, the dyes are absorbing the red and the blue color light, as we can appreciate. So these two color of light, okay, they are getting absorbed. Okay, whereas the green light, if you see, it is being emitted by the dye, and that is actually the color of the dye which is visible to the naked eye. Okay, remember, a particular chromatophore has multiple electrons which is helping in absorbing light of particular wavelength, and not only that, these many free electrons are also helping in absorption of the UV rays which are out of the naked spectra of the eyes. Okay, so this is all about the chromatophore. Then we are having the oxochrome. As I told you, this part of the dye is helping to intensify the light. Okay, it is an ionizing group that also helps to stick the stain to the particular tissue. It is giving more free electrons to the chromatophore group to facilitate more light absorption, especially light of a longer wavelength. So as we can appreciate over here, we are having a chromatophore and we are having an oxochrome, which are the two components of the dye. The chromatophore is helping in absorption of light of a particular wavelength, and it is emitting rest of the light, which is giving the color to that particular dye, and the oxochrome is the component which is helping the chromatophore to absorb more light, thus increasing the intensity of the light. Okay, I hope this portion is clear. Now we will try to understand what are the various types of dye. Okay, so dyes can either be anionic or acidic dye, cationic or basic dye, neutral dye, ligand or chelating dye. So what is an anionic or acidic dye? So these are, as the term suggesting, these are negatively charged. Okay, so they are negatively charged. Okay, so they will therefore bind to the positively charged component of the cell, that is, they will bind to the cytoplasmic proteins and the collagen which are positively charged. And what is the example of acidic or anionic dye? The example is eosin. Okay, So this is about the anionic or acidic dye. Then we are having cationic or basic dye, which are positively charged. So they will bind with the negatively charged components like the nucleic acid and the epithelial mucin. So what are the examples? Methyl and ethyl green and Alcian blue. These are examples of your basic or cationic dye. Then we have neutral dye. So very simple. You mix acidic along with the basic dye. The mixture is called as a neutral dye. So they are going to stain both the positive and the negative components in the cell, that is the nucleus as well as the cytoplasm. The best example of this is the GMSA stain. Okay, the GMSA stain. Okay, then we are having the ligand or chelating dye. Okay, these are actually weak acids. So more or less you can tell that these are anionic or they are negatively charged. Again, they are also staining the nucleus and the cytoplasm. The best example of these ligand and chelating agents, these are aluminium hematoxylin, examples being Harris hematoxylin or Mayer's hematoxylin, and iron ligand with hematine, that is iron hematoxylin. So these are basically your ligand or chelating dye. So these are the different types of dye. So now we are just going to read whatever we have just summarized right now, and we are just going to add few more points. So as I told you, the anionic dye or the acid dye, they are carrying negative charge and they will migrate towards the anode in an electric field. They are mostly low molecular weight. They are very importantly soluble in water. Okay, so most of the dyes, they are having 2 or more anionic groups which are making them soluble in water. This is an important point to note. These dyes will combine with the tissues that carry positive charge. Okay, and those tissues are called as acidophilic. So what are these components? The cytoplasmic proteins and the collagen. Most important example over here is the eosin. It is one of the most commonly used acidic or anionic dye. Okay. The next is your cationic dye or the basic dye. They carry a positive charge. Okay, and they move towards the cathode in an electric field. Now these are mostly soluble in ethanol. This is again an important observation you have to make. So these basic dyes, they will combine with the tissues which are carrying negative charge. What are these tissues? Basically the nucleus. Okay, basically as we have seen, the nucleic acid and the epithelial mucin. Okay, this negatively charged tissue combining with the basic dye is called as a basophilic tissue. Okay, what are the examples? Methyl green, ethyl green and Alcian blue. Then we have neutral dye. So these are compound dyes that contain both acid and basic dyes in combination. So in aqueous solution, the acid and basic dyes will exchange electrons and they combine together to precipitate in the tissue. So Romanovsky GMSA staining is the best example of the compound dye that undergoes this electron transfer process. Now remember, this GMSA has 2 components. One is the azure B and eosin Y. Now the azure B dye, it is an electron acceptor, whereas the eosin Y, it is an electron donor. So there is a transfer between these 2 components and they neutralize each other. So the example is your GMSA stain. Then we have the ligand or the chelating dye. So the ligand dye, it is the complex compound that consists of dye as well as a metal ion. They are also known as metallochrome. They are usually weak acids, as I have already told that they are weak acids. They are anionic in nature. Okay, so they are consisting of a dye component and a metal ion. Okay, so hematine, it is oxidized hematoxylin and it is used as a combination of either aluminium ions or iron ions. The metal ion dye complex, okay, has surplus charge that increases the solubility in water and makes the dye insoluble in alcohol, and therefore dehydration due to ethanol does not occur during staining. So it is very important that this complex, that is the dye metal ion complex, have an excessive amount of charge, is increasing the solubility in water, and therefore what is happening, that it is making the dye insoluble in alcohol. That is why during the dehydration process, it is it is basically it is resisting the dehydration process. Okay, it is therefore dehydration due to the ethanol does not occur during staining. Okay, remember, because the dye becomes soluble in the water. Okay, and therefore that dehydration process due to ethanol does not occur during the staining, thus retaining the dye. So the 2 examples are aluminium hematine and iron hematine. Now aluminium hematine, it is a type of ligand dye where the metal aluminium is combined with hematine. It is used in Harris hematoxylin and Mayer's hematoxylin. As aluminium, this particular metal and dye complex, it is insoluble in ethanol. So it can be used along with other anionic dyes, and both dyes are retained even after dehydration with alcohol. That is why they are used for staining. Then we are having iron hematine, that is Fe+2 hematine. It is the ligand dye that consists of iron and hematine. The dye is made by combining iron salt and hematoxylin. Iron binds with the hematoxylin molecule by oxidizing it to hematine. Iron hematine is used for staining myelin and elastic fibers. Okay. Now, very importantly, we are going to discuss about the various mechanisms and theory of staining. Again, I am repeating, this is a very important concept to understand, and a lot of examiners are asking this as the first line of question. So, if you are not able to answer such simple basic questions, your Viva is not going to proceed any further. So, basically, the staining is the combination of a colored substance, that is the dye, with a particular tissue that is retaining the dye even after washing. The staining is primarily a chemical reaction between the dye and the tissue. The following chemical reactions are involved between the dye and tissue components, which we are going to discuss individually. So what are those? Let me enumerate. We have electrostatic bond, van der Waals attraction, hydrogen bond, covalent bond, hydrophobic bond, dye aggregation. So these are the different types of bond which are available. So let us see the first important bond, that is the electrostatic bond, also called as the Coulombic forces. Now such kind of bond is existing between two oppositely charged particles. That means the dye is having one charge, tissue is having opposite charge, they are binding with each other. Very simple to understand. So the concept is exemplified as I have already mentioned. So this is a particular cell. So if there is an acidic or anionic dye which is negatively charged, so they are going to go and bind with the tissue components which are positively charged. What are these tissue components? The cytoplasmic proteins as well as the particular collagen. Okay. Similarly, you can have basic or cationic dyes which are positively charged. So this positively charged basic cationic dye will go and it will bind with what? With the negatively charged component. What is the negatively charged component? That is the particular nucleus and even the acidic mucin. Okay, so this is how electrostatic bond or Coulombic forces are generated between these two dye tissue complex. So dye, always remember, a particular dye is comprising of a chromogen and oxochrome, as I already told you, and these two components are oppositely charged. Now when the dye is basically mixed in a particular solution, okay, in the solution form, they will dissociate. So for example, if we are using a basic dye, so in a solution the basic dye is going to break apart. Okay, it will break into chromogen, that is the part which will give the color, and into an oxochrome part which is negatively charged. So they will break apart. So this positively charged, now because we are using an acidic dye, so the chromogen will be positively charged. They will bind with the negatively charged tissue components like the nucleus and the acidic mucins. Similarly, the acidic dyes, the acidic or anionic dyes like eosin will combine with the positively charged tissue components like the cytoplasmic proteins or the collagen. So the best example of a dye which is following or which is having, electrostatic bond or Coulombic forces, we can give one acidic dye, name an acidic dye like eosin. So this is the first theory, that is the electrostatic bond or the Coulombic forces. The next very important variety is the van der Waals attraction or the van der Waals forces. Now these are weak non-Coulombic forces. In fact, it is the weakest force due to intermolecular interaction. So what is the principle of this attraction? Remember, when the electron of an atom is concentrated at one pole, then a dipole is formed. Okay, and this is very much reminiscent of magnet having two poles. So very simple, this is one particular atom. So what has happened, that if one pole, this is one pole, this is another pole. So if one pole is having very less electron, another pole is having excess amount of electrons, so they form two different poles, and that is when you call them as a dipole, because they are having two poles. This is one pole having less electrons, which is another pole having more electrons. Okay, so this is basically having two poles. So the van der Waals attraction, there can be of three main types. One is the dipole-dipole interaction, dipole-induced dipole interaction, and dispersion or London force. So let us look at the first important interaction, that is the dipole-dipole interaction. So what happens over here in the dipole-dipole interaction, if you see, the bond is there between already formed two permanent dipoles. Okay, so let me give you an example. So over here, if there is a marriage, between the husband and the wife, and over here both husband and wife, they are already established doctors, so they are basically permanent dipoles only. Okay, so basically over here one permanent dipole which already is in this particular configuration, okay, is getting attracted or interacting with another permanent dipole like over here, okay. So basically this part which is having more electron is getting attracted to this part having less electron, okay. So they are having an interaction with each other. This is called as a dipole-dipole interaction, and it is between two permanent dipoles, already established dipoles, okay. The second variety is dipole-induced dipole interaction. So over here, the husband is marrying a wife. So husband is a doctor, wife is yet not anything. She is not doing anything. After marriage, the husband is, inspiring the wife to give the exams and to become a doctor. So over here, the husband is inducing the wife to become a doctor. That is, this is called as a dipole-induced dipole interaction. So over here, the bond is there between a permanent dipole, a dipole which is already existing. Okay, so and this particular end is actually inducing. So this particular dipole actually had equal number of electrons. Okay, but because this particular end came close to this end, so many electrons they migrated towards this part, and only one electron was left over here. So over here the interaction is there between one permanent dipole and an induced dipole. Such kind of interaction is called as a dipole induced dipole interaction. Okay. Thirdly, we are having a dispersion or a London force. So what is over here? Let us try to understand. So a permanent dipole, a person who is already established. Okay. So a permanent dipole is already existing, and they are basically inducing another dipole. So this group of electron is interacting with this. So this wasn't a permanent dipole. This was a normal dipole, and it was induced. Okay, it was induced to become a dipole. So basically a permanent dipole is interacting with the involved dipole. Now what happens that this involved dipole, this induced dipole further induces other dipoles. Okay, and there is a plethora of induced dipole interaction. This is called as dispersion or London force. So over here the induced dipole further induces a chain of induced dipole. So this causes a large network of tissue may undergo induced dipole interaction in such a manner, and this is actually the basis of dispersion or the London force. Okay, I hope this portion is clear to everyone. So what is the example of a stain following this principle or this particular Van der Waals force? We are having the elastin stain by Miller stain as well as the Congo red stain. These are examples of Van der Waals interaction. Now there are different other kinds of bonding also. So what are those? They are hydrogen bond. Now hydrogen bond, it is a weak bond type of a covalent bond which is occurring between the hydrogen and a